A very good morning to all of you and welcome to this webinar on technological challenges during the COVID pandemic. And we have a very eminent panel of speakers. And I should also say good evening to one of our resource persons, Dr. Prabhakar, who is joining us from Chicago. He is the Dean of Medical School in Chicago, University of Illinois. And we are having another expert, a nationally and internationally well-known virologist of the country, leading virologist, Dr. Ravi. He will be joining in a couple of minutes. And we also have Dr. Gangadharan from the National Institute of Technology, Suratkal. So he is one of the resource persons. And our Honorable Vice Chancellor, he will be giving the opening remarks. So we are very happy to welcome all of you for this webinar. Today, we are in the midst of coronavirus pandemic that has affected more than 5 million people and has caused more than 300,000 deaths globally. And we seem to be at crossroads, and most of these seem to be in countries that are resource rich and which have a very good public health system. So what are we lacking? So we seem to be at crossroads despite all the scientific and technological advances today. Now, it may sound strange that if you look at the history of virus diseases, Edward Jenner developed the concept of vaccination even before viruses were discovered. So in the 18th century, he developed this concept. At that time, we didn't even know that germs exist and they cause the disease. And then even Louis Pasteur developed the rabies vaccine even before the virus was discovered. So we have this history of trying to overcome the problems. And even we talk about the Spanish flu, which happened in 1819. But even at that time, it was not known, and the virus was discovered only in 1931. And the first human virus to be discovered was in 1901, the yellow fever virus. And since then, we have made significant advances in the knowledge regarding viruses. And today, with all the genomic data, it indicates that human body has more than 2,500 viruses. And more than 200 viruses can affect humans. And today we have technology for growing viruses. We have technology for sequencing, developing diagnostics. Now, despite all these technological advances, we seem to be struggling to control this pandemic due to coronavirus. Though, of course, coronaviruses have been known to cause some of the respiratory illnesses before. This is not the first coronavirus to be discovered, but still we are struggling to cope up with this pandemic. So at this time, I think we need to look back and see where we are missing. So this shows that despite all the technological advances, we still have a lot of things to learn, and a tiny microorganism can teach us a lot. And we have forgotten many things about these microorganisms. So in this context, our Honorable Vice Chancellor considered it important to have a webinar inviting global experts to discuss the technological advances and technological challenges that we are facing today. So I would like to now invite our Honorable Vice Chancellor to give his welcome remarks.
Thank you, Vidya Karunasagar. On behalf of Nitte, I welcome the distinguished panelists, Dr. Bellur Prabhakar, the son of the soil, our good friend from USC Chicago, Dr. Gangadharan, the National Institute of Technology, Karnataka, Dr. Ravi, virologist from NIMHANS, Bangalore, for this very important webinar on technological challenges during COVID pandemic. As Vidya said, we are in the midst of global public health crisis, according to me. COVID-19 has exposed our shortcomings in the preparedness to meet the challenges posed by viral diseases. In spite of all these massive developments in healthcare technology. Now we have the now we have realized the COVID-19, it's going to remain in the community for long, and we have to live with it. Many more such viral infections may emerge in the future also, and our governments have to be prepared to face the challenges, and the countries should develop robust strategy to prevent, contain, and treat these infections by using modern technology. Fortunately, we are in this millennium, with all the developments, we are better equipped to handle such pandemics compared to 1918. What India said, those days we had 500 million people got infected and more than 50 million, I think, might have died in this pandemic. But we are better placed now. Thanks to the application of modern technology, particularly in educating the public, surveillance, contact tracing, and diagnostic tests, modern intensive care equipments. You'll all agree with me that now the things have changed, you are better placed, and you will be surprised to know that India has done so much during this pandemic, and we have used drones to manage lockdown period. That you will be surprised that probably many countries would not have ventured this type of technology to even manage lockdown and to make the people to stay at home. You may be knowing that Indian government has started an app called Arogya Setu, which is probably one of its kind in the world, and that is to trace the contact tracing. It has helped the government in contact tracing. In our university, as I was talking to Prabhakar, we have done our bit. Particularly when we went for lockdown, we were worrying how to engage the faculty to work from home. Probably it's a new culture for Indian universities. And we could engage them in online teaching and also assessment by providing our own in-house new learn technology, which has been developed in the university by efforts of a lot of people, faculty, and it has been well used during this period, and we could conduct teaching and assessment. And as I said earlier, we used telemedicine facility to treat the patients, not only from our district, from other states also, even from abroad. And necessity is the mother of invention. I am proud to tell you that Indian scientists are working overtime to deliver cost-effective diagnostic kits, PPEs, ventilators, drugs, and vaccine to mitigate the human suffering due to COVID-19. To discuss these technological challenges, I think we have got very eminent panelists, and they have been introduced by our friend Vidya Karunasagar, Dr. Belur Prabhakar, Dr. Ravi, and Dr. Gangadharan. Prior to this pandemic, our diagnostic, but that one more achievement, what we have done recently is we got the permission to start our virology lab. Dr. Ravi is the nodal officer from Karnataka. He was instrumental in facilitating the arranging the COVID lab in the hospital. I welcome him for this webinar. And I welcome Professor Dr. Prabhakar from USC Chicago to this webinar, and definitely his valuable thoughts and suggestions. What they did in USC Chicago will be very valuable, and it will enlighten the participants. And we have got Professor Gangadharan from NITK Surathkal, and he is the person handling technological inventions rather, like ventilators and other facilities to be created for managing COVID patients. His inputs will be very valuable. I hope there will be very interactive, a very informative webinar concerning the technological challenges faced by people in managing COVID. Welcome you all once again for this webinar. Thank you. Over to India, Karunasagar.
Oh, so okay, thank you very much. And yeah, now I see that Dr. Ravi has just joined. So welcome, Dr. Ravi, and we started the introductions, but now I think Indrani will introduce Dr. Ravi to the group. Thank you.
The topic for today's webinar is a challenging one. As the title suggests, technological challenges are going to be addressed by speakers who have carved a niche in their own chosen fields. We know the lockdown in the country enabled preparedness for whatever is in store, and therefore, when we look at COVID-19, which has led to very unprecedented times, what we really see is the number of questions that come in the minds of people. And first and foremost is looking at diagnostics. Who else than one of the country's leading diagnosticians, leading researcher, and when we have all in one, a clinician, a researcher, and someone who does work at ground level addressing the problems that have arisen in the minds of people, we could not have thought of anyone better than Dr. Ravi. In looking at tests, people have always wondered what kind of tests need to be done if one is asymptomatic, if one is symptomatic, if one is looking at community profiling, surveillance programs, and if any test is to be done, what does the test mean at all? Because overdiagnosis, fright, panic can all lead to the kind of problems that we have been looking at. The stigma that we speak about is because of the intense fear and the panic that has come about. Nevertheless, the government has addressed all of this through the efforts of people like Dr. Ravi. Without much ado, I should now introduce this eminent speaker, a senior professor and head at Neurovirology Department of NIMHANS now, a registrar formerly of the same institute, and one who has passed out from very reputed institutions like JIPMA in Pondicherry. One expects that this country at these unprecedented times has the best of people leading her in managing a problem that the whole world is struck with. Apart from the fact that he joined as a young boy as a lecturer in the newly formed Neurovirology Department at JIPMA, at NIMHANS, when the department was first, I think, inaugurated by a Nobel laureate, as I see from the reviews that I've read about this. He rose to the ranks of a professor, and since 1995 has been managing the department extremely well. Being a good scientist, being a good clinician, managing a department is one part of it. Being a trainer, helping the country, the state to move forward, has rightly enabled him to be chosen as the nodal officer for Karnataka by the Indian Council of Medical Research. He has been at the forefront of training several laboratories and Indian Council of Medical Research, realizing that a few labs are not enough for a country as vast as ours to manage issues of testing, has brought him to do this arduous task of training almost every medical college that desires, after setting up the virology laboratory, gets an ABL accreditation. And in all of this, we are no exception, and Nitte is highly indebted to Dr. Ravi for the support as he has given for everyone else. Needless to say, we are very, very proud of this gentleman in Bangalore, who is also a fellow of the Academy of Medical Sciences. Thank you, Dr. Ravi, for so kindly accepting to take on diagnostics and testing as a topic in which I'm sure there will be many questions. We may not have time to address all of them at the end, but you would be there, always available for anybody to get in touch with you. Thank you for coming over. Thank you.
So over to Dr. Ravi now. Dr. Ravi, I think your mic is muted. Your microphone is muted. Dr. Ravi, your microphone is muted. Your microphone is muted.
Your host will be able to unmute him. Mahesh can unmute him. In fact.
Yeah, now I am unmuted. I'm sorry. Good morning, and let me thank Professor Karuna Sagar and Dr. Indrani Karuna Sagar for having me on this webinar. It's a great pleasure. I visited Nitte during the virology conference, and I want to say that I'm highly impressed with the kind of work that they are doing. What I will do in the next 20 minutes is to try and talk to you some of the challenges we have faced in the diagnostics of COVID-19. Can I request my slides be projected, please? I have sent it on email. I'm unable to do so from my phone. So if somebody can project my slides, it will be very useful. While the slides are coming up, let me just tell you what I intend to do in 20 minutes is to give you a brief overview of the virus, the most important structures of the virus that are used in diagnosis and in immune response, the kinetics of the immune response, the clinical spectrum of COVID disease and when we do what, laboratory methods, and finally the limitations. Can it go to full screen mode, please? Hello? Can it go to the full screen mode? Hello? Thank you. Can you go to the third slide, please? The next and the next. Yeah, thank you. This is a slide that is showing you the structure. One slide back, please. The structure of SARS-CoV. It's an enveloped virus. I'm sure all of you know that it's an enveloped virus. It measures anywhere between 60 to 200 nanometers. It's a RNA virus. The most important protein of the virus is the surface glycoprotein known as the spike protein, which is immunogenic, which is responsible for attachment to human cells. So biologically, it's a very important protein. By the side of the spike protein is situated the envelope protein, which is also immunogenic, and which is one of the targets that is used in the real-time PCR diagnostic tests. And surrounding the nucleic acid is the nucleoprotein, which is a very important protein again in diagnostics. People are using that as one of the targets. The genome itself is about 27 to 30,000 kilodaltons. It's on the right. You can see it has both structural and non-structural genes. Coronaviruses usually have open reading frames, and the SARS-CoV-2 or the COVID-19 virus as it is called, SARS-CoV-2, has two open reading frames, 1A and 1B, and the 1B and 1A code for some unique proteins of SARS-CoV-2, one of them being the RNA-dependent RNA polymerase, which has very unique sequences compared to the other coronaviruses. So basically, I put up this slide to tell you that in the PCR that we are doing today, various companies have targeted spike protein gene, envelope protein gene, nucleocapsid gene, as well as the RNA-dependent RNA polymerase gene, and some of them unique sequences in the open reading frame. Next slide, please. SARS-CoV-2 causes a wide spectrum of illnesses, ranging from asymptomatic, which should be actually on the left in the spectrum, to very severe pneumonia and critical illness and death. So in between, you have a range of presentations a SARS-CoV patient will have. It may be mild fever, it may be just fever and cough, shortness of breath. Some people present with diarrhea. Fatigue is a very well-known symptom in SARS-CoV. And of late, people have described, and even CDC has revised its symptom list to include new onset of loss of smell and new onset of loss of taste. So these are the wide, and of course, pneumonia is one of the hallmarks as it produces a severe acute respiratory syndrome. So these are the wide range of clinical manifestations that you see in SARS-CoV-2. Next slide, please. Before we go into the challenges and what the diagnostics are, I thought we should talk about the kinetics of what happens in a SARS-CoV-2 infection. This is, at this point of time, a hypothetical model that has been constructed. This is a new virus. All of us are learning, and there are several limitations in the way we learn. The graph tells on the x-axis the number of days, and on the y-axis the level of a particular parameter that is being measured. If somebody gets infected on day 0, usually symptoms, median time for symptom onset is 5 to 7 days in 80 to 90% of COVID-19 patients. Prior to that, they will be asymptomatic, and some of them continue to be asymptomatic for quite a long time. We are just beginning to understand this. But if careful history is taken, there will be some mild symptoms. So what you read in the press that 80% are asymptomatic is actually not correct. If somebody sits with the patient and goes through very meticulously, the number of people who have mild symptoms will be a large chunk within this so-called asymptomatic. So during the first 2 weeks, there is virus multiplication in the respiratory system. It can spill over to the blood, but it's usually not a common feature. But the virus multiplies in both the upper and the lower respiratory system. So viremia or virus detection can happen up to 2 weeks. In some patients, it can go very rarely beyond 3 weeks and 4 weeks, but those are exceptions. 80% of the people clear the virus by day 10 or day 14 maximum. Antibodies start appearing around the end of first week. IgM is the first to appear. And then IgM lasts for usually about a month to 3 months. IgG starts appearing beyond 2 weeks. Unlike most other acute viral infections, the immune response window is shifted towards the right and it's delayed. In most arboviruses, you will start seeing antibodies appearing on 3rd day, 4th day after infection. But here, you see IgM itself appears at the end of 1 week and IgG beyond 2 weeks. I'll come to this when I talk about antibody detection test. Next slide, please. In SARS-CoV-2, the laboratory is called upon for a variety of reasons, which Dr. Indrani was mentioning. One of the major reasons is to diagnose the infection, but diagnostics are very useful for surveillance, both in terms of finding out exposure in the population to the virus, how many people have got exposed. And also, diagnostics are today used for staging. In the clinical staging they are used. Some of them have been used as prognosticators. Some of the markers that people have found, like D-dimers and elevated levels of cytokines, especially C-reactive proteins like C-reactive protein, procalcitonin. These are some of the therapeutic monitoring or indicators that have been proposed in SARS-CoV-2 infections. So the laboratory plays a very important role in various stages of diagnosis. Next slide, please. What are the indications for doing a COVID test in India? About 4 days back, the Indian Council of Medical Research revised its guidelines for COVID testing in India. Earlier, they used to have many other criteria. Now it's simplified. Basically, the sum and substance of this 9 points that you see here is a test should be done primarily in those who are symptomatic. What symptoms? Influenza-like illness or severe symptoms of acute respiratory infection like shortness of breath, pneumonia, admission to an ICU, and so on. We are moving away from testing everybody in the community to focused symptomatic testing. Ultimately, as a disease starts spreading into the community, it's impossible to test everybody because we do not have the resources. Focus should be on those who are symptomatic. There is also evidence now accumulated, and you can see this on CDC website, as well as India's discharge policy, which many people threw their hands up and shouted, how can you discharge a patient without doing a test? There is enough evidence now globally that if people are asymptomatic, they are admitted with symptoms, whether mild or severe, once they do not have symptoms for 3 to 4 days, you can discharge them without doing a test. That is the current guideline. Because the test that we use detects viral nucleic acid. The test that should be used ideally in every patient is virus isolation. There's a world of difference between virus isolation and detection of viral nucleic acid. It has been found through various epidemiological studies now that the moment people do not have symptoms for 3 or 4 days, they are not infectious within inverted commas. They are not infectious, so they can be discharged. So there's a lot of hue and cry about asymptomatics going around in the community spreading. Actually, there is very little hardcore evidence published in literature on that. People detect a number of people who do not have symptoms who are positive. But the so-called super spreaders, there was a paper 2 days back in Science as well, are those who are always symptomatic, not those who are not asymptomatic. Next slide, please. So what test do you do and when do you do it? The test that is recommended for diagnosing COVID-19 disease today is the real-time PCR. It is best done within the first 2 weeks after onset of symptoms or after contacting the disease. After that, the sensitivity of the test will go down. Beyond 2 weeks, its antibody tests are useful, but by that time, diagnosis is already made. So antibody tests are not diagnostic tests. They are tests to find out if somebody is infected with the virus or had past exposure to the virus. And at best, if you do neutralization, you can say whether the person is protected. Next slide, please. The specimens that are used for confirming diagnosis is the nasopharyngeal swab. On the right, you can see that You have to position the neck of the patient at 70 degrees, comfortably seated on a chair, and gently push the swab to the back. One indication of proper collection of specimen is the person will feel uncomfortable and usually raise their hand or tilt their neck backwards. That means you have touched the nasopharynx. The second specimen is a oropharyngeal swab. Use an ice cream stick, depress the tongue, explain the procedure, gently roll the swab on the posterior pharynx. How do you know you've reached the pharynx? The person will gag. So these are two indications. If the specimen is not collected properly, you are going to give a wrong diagnosis. Next slide, please. So nucleic acid tests are the current recommended tests. If you ask me what is the gold standard in SARS-CoV, the honest answer is there is no gold standard. Virus isolation remains the gold standard. It's not practical because you need a biosafety level 3 lab to handle specimens and grow virus. So nucleic acid is the next best choice that we have. It detects current COVID infection. The utility of the nucleic acid test is to inform the individual that he has an infection, to inform the public health system that this is a person who probably can spread infection to others. So if necessary, isolation has to be done so that we can prevent transmission. The individual benefits by knowing the diagnosis, although I must say that 80 to 90% of them will not even require hospitalization because they have such mild symptoms or no symptoms. So that is the utility of the nucleic acid test. Antibody tests, I already mentioned to you, says past exposure to virus. A negative test, if done beyond the 14 days, is very valuable to say that I have not been exposed to SARS-CoV. A positive test definitely is useful to say I have been exposed, and if the functional activity of the antibody has been measured through neutralization, you can say the person is protected. Next slide, please. Next slide. You can skip this, go to the next one. Essentially what I told in the previous slide. Next slide, please. Yeah. This slide summarizes antibody testing. You today have a large number of, go back please, one slide. We have a large number of antibody tests that are available. There's aggressive marketing of the rapid kits for IgM and IgG. By large, the performance of rapid kits has been below par, much below the claims of the manufacturers. We have had experience with about 6 rapid kits so far. The claimed sensitivity is 90% of subjects will have antibodies 14 days after they have turned positive for SARS-CoV by RT-PCR. We have a panel of 45 such samples, and we have a panel of about 65 negative samples. Almost all the tests, all the 6 tests that we evaluated had a sensitivity of between 40 to 45%. That means less than half the number of confirmed patients beyond 14 days were positive in the rapid tests. Our limited experience with ELISA has been very good. This is an ELISA developed by Dr. Gagandeep Kang from the Translational Science Health Technology Institute in Faridabad. And you also know that the National Institute of Virology has made India's first indigenous ELISA kit. Almost all ELISA kits use the spike protein, especially the receptor binding domain within the spike protein, which is most antigenic and which is very specific to SARS-CoV-2. So that is coated on the plate. And people have developed now IgA, IgM, and IgG assays. Very recently, the controller has licensed many kits. They are available in the market. Even chemiluminescence kits are available. Neutralization remains the gold standard among antibody tests to measure protection. Unfortunately, you need either chimeric virus and also a biosafety level 3 facility. Lastly, I'll wind up by telling you the limitations of diagnostics. Next slide, please. The limitations in the current test that we are using, the biggest limitation is the type and the quality of the specimen that is collected. Quite often we have seen some of the samples failing RNA-sp, the housekeeping gene, which is amplified in a real-time PCR, because people do not collect nasopharyngeal and oropharyngeal swabs properly. So specimen collection issues limit diagnostics to a great deal. Second thing is, both nasal and oropharyngeal swabs have to be collected from one patient and put into the same virus transport medium. People are still learning to do this. Why you have to collect two specimens and put into one BTM? The answer is the sensitivities for the nasopharyngeal swab and the oropharyngeal swab is different. There are a few studies which have said nasopharyngeal swabs are better than oropharyngeal swabs, but I must tell you the jury is not out on it yet. But we know if we combine both nasopharyngeal and oropharyngeal swabs and put it a VTM, by 5th day after infection, the sensitivity is 90% for real-time PCR. If you collect a specimen before 5th day, your sensitivity will fall. If you collect a specimen beyond the 10th day or the 12th day, your sensitivity again is likely to fall. So it all depends on the right time when you collect the specimen, and the correct specimen corrected properly is the biggest hurdle to using RT-PCR. So one has to get that very clearly studied. Next slide, please. As far as antibody tests go, the limitations are that it cannot diagnose a current infection. Most people who have used rapid and ELISAs have found that IgM responses are rather poor in COVID-19 cases. We do not know why. Unlike arboviruses, where there's a brisk early IgM response in COVID-19 is not brisk, nor is it very early. IgG is a much better antibody to detect, but by the time IgG is detected, it has no diagnostic significance. I'll stop here and thank you for giving me an opportunity to speak on this webinar.
So thank you, Dr. Ravi. I think a lot of participants will have a lot of questions, but we can take it up later in case we have time. Now we will proceed with the talk from Dr. Prabhakar. Dr. Belur Prabhakar, I've been fortunate to be his classmate during his master's degree at the Kasturba Medical College, Mangalore. We did MSc Medical Microbiology together there. And then he moved to the United States, where he did his PhD from Johns Hopkins University, Baltimore, and then did his postdoctoral studies at Pennsylvania University, and moved to the National Institute of Health, where he got elevated to senior investigator. He then moved as professor to the University of Texas Medical Center. And then since 1997, he's at the University of Illinois as the professor and head of microbiology and immunology, and presently he's also the dean for the medical school. So we are very fortunate to have Dr. Prabhakar here, and he will talk to us about vaccine. Of course, everybody is hopeful that a vaccine will come up soon. So he will talk a little bit about the immune response and the potential for vaccine development. Over to Dr. Prabhakar.
Yeah, thank you very much, Dr. Sagar. Dr. Ravi, virtually I'm going to say hello to you.
Hi.
Not only we are kindred spirit in interest in virology, but we also have goatees. This is my COVID beard. So, Sagar, if you would be kind enough to show my slides, please. And Dr. Ravi has set up a beautiful introduction, which actually allows me to very nicely dovetail into his presentation. And let me have the slides first. Dr. Gangadharan, I should also tell you, my nephew finished his engineering college at Suratkar Engineering College.
Oh, that is very nice to hear. Which year?
Oh my God, his last name is Ashwin Bellur, and he finished that, and then he went to IIT Madras, and now he's doing his PhD at Johns Hopkins.
Yeah, good to know that.
Yeah. Sagar, still I don't have the slides. I would say he graduated maybe 8-9 years ago, something like that.
Surely he should be knowing.
Yeah, I'm sure he will.
Hello, Dr. Ravi.
Chairman.
Welcome to the webinar. We missed you in the initial introduction time. Welcome to Nitte University webinar, and thank you very much for mentoring us to start our COVID diagnostic lab. Yesterday we got our NABL accreditation.
Today you will get in your login and ID credentials and you can start. By afternoon you will get.
Thank you very much.
Dr. Ravi, were you involved in helping Ramaya Medical College as well?
Yeah, they are still in the process of getting.
Yeah, they're still in the process of getting their biosafety cabinet.
Yes.
So you are working with, Gangadharan is working with them now. Pratap is working with you, correct?
Yeah.
Yeah, they talked to me a couple of times.
I see.
We can have some questions to Ravi.
Dr. Ravi, yeah.
Yeah. One was a surgeon. I think we must have, during last interaction also, you must have enlightened surgeons. Even yesterday we had discussion whether we should have a diagnostic test of COVID before venturing elective surgeries.
My answer would be that you are the best judge. Having said that, I will say that I've pointed out all the limitations of testing. So keep that in mind. To do a test or not do the test is the prerogative of clinicians. But the challenge is, if you have tested at the wrong time and you got a negative result, you will presume patient is negative. But post-operatively, if he's in the incubation period, he can manifest symptoms, and that can be quite disastrous because at that time you would not be placing him in a COVID ward. So all laboratory tests have limitations. I am yet to see a test that is 100% sensitive and 100% specific. So I would like to draw the analogy of HIV, saying that presume everybody is infected and take appropriate precautions. Over.
Thank you.
Yeah. Beautiful. Yep. Okay, Sagar, can you go to the next slide, please? I think we can even skip the next slide because Dr. Ravi has beautifully. Okay, so this is the current situation in a snapshot. This is the data from 4 p.m. this afternoon, and this is the Johns Hopkins has a COVID information live. I mean, literally, you can watch the numbers change while you are logged into this space. And at the end of my presentation, I will give you the login information for all of these websites. So as Dr. Ravi mentioned, we have over 5 million cases and nearly 330,000 people around the world have died. And the graph at the lower right-hand corner is basically the trend line. If you look at the trend line, it has not flattened. It continues to grow. Part of the reason is because I think more and more people are being tested, and that is probably one of the major contributing factors. But also social distancing throughout the world, almost throughout the world, had definitely slowed the spread of the virus. But as they are lifting the social distancing or the stay in place rules and regulations, it is anticipated this curve will continue to go up for quite some time before it settles down. Can I have the next slide, please? And I just want to show, this is I pulled out the data exclusively for India. As you can see, as of April first week, the cases started showing up in India, and as of now, we have well over 100,000 cases in India, and all of you are acutely aware of the mortality associated with these cases. And India was one of the countries that implemented stay-in-place policy quite early and quite aggressively. So that might have really helped slow down the spread of infection. But as the India begins to reopen and people start getting back to their quote-unquote new normal life, it is anticipated these cases will continue to rise. But how long it will continue to rise, we don't know. Only time will tell. And the bottom gray line is the mortality. As you can see, case mortality rate in India is quite impressive, given all the challenges the country has in terms of inadequate healthcare, challenges with social distancing, challenges with personal hygiene, and so on and so forth. In spite of all of that, I agree with Dr. V and Dr. Bhandari. India has done remarkably well so far, and let us hope and pray it will continue this way. Can I have the next slide, please? So I deliberately have put this, this is one index case. This happened to be a German guy, who happened to be in China. And you can see, he visited Germany, and then he takes a flight to, this is a Chinese guy from Wuhan. And you can see the symptoms appeared on around January 22nd, because he was there in, attending some business. And his PCR result appeared, and it turned up positive on January 26th. So it took about 4 days from the onset of symptoms by the time the person was tested by RT-PCR. And then you can see the index case is there, and then you will see patient 1, 2, 3, and 4. And what I would like to impress upon you is that how quickly the symptom goes from, for example, this for index case developed symptoms approximately on January 22nd. The next case appeared on January 26th. The third case appeared on January 26, 26, 27, by 28. Within a matter of 5 to 6 days, he had spread the infection to 4 other people, and they all showed up PCR positive by day 28. So the point I would like to impress upon you here is that one index case very rapidly was able to transmit the infection to 4 other cases, and you can only imagine if these 4 patients came in contact with 4 or 10 or 20 other people, how rapidly the virus could spread. This is literally what happened in the early stages of COVID breakout in Wuhan, and then the same thing happened in New York City, same thing happened in Detroit, same thing happened in Louisiana. Looks like it took the world quite a bit of time to really appreciate how quickly the virus can can be transmitted from one person to another person. Can I have the next slide, please? And I'll tell you why this is important, because this helps us, design our public health intervention as well as, vaccine and drug development strategies. So Dr. Ravi has already very clearly articulated how COVID-19 is diagnosed, and it's primarily by RT-PCR as of now. And when we look at RT-PCR, we are not looking at live virus. This is another myth that many people fail to understand. RT-PCR simply detects viral nucleic acid. It doesn't even have to be full-length viral nucleic acid. All it has to is have a piece of viral nucleic acid to which the primer binds, and then the RT-PCR will amplify that. So therefore, we are looking for not necessarily live virus. We are looking for evidence of viral infection in the individual. The virus may be alive, virus may not be alive. However, the RT-PCR can pick both of those up. So RT-PCR result does not necessarily indicate that the patient has live virus. As he clearly mentioned to you, the virus isolation will be the gold standard for that. And also, he already talked about the limitations, so I want to belabor the same point. And also, what are the serology tests and how are they used? The serology tests help identify previously infected individuals, as Dr. Ravi very clearly mentioned. And also, it might be helpful in identifying people who are asymptomatic carriers because they may have mounted an immune response, and yet they had no signs or symptoms of COVID-19 infection or symptoms, and therefore we could identify people who are asymptomatic carriers if they were to mount an immune response by looking for antibodies. So Dr. Ravi has already told you why there are so many inconsistencies among testing data for COVID-19. First and foremost, I don't know about in India. In America, the way the results are reported itself, causes confusion. For example, they will only tell you how many people they tested and how many were positive. And sometimes they will simply come at the end of the day and say, today we had another 150 positive cases. So the cases keep on accumulating without denominator. Those 150 cases really become less meaningful in terms of understanding what's happening in the community. And of course, many a times, the same individual might have been tested for more than once. So they report number of tests, but not the number of individuals many times. So therefore, there is a lot of internal inconsistency in the way the data are reported. And also that leads to some confusion about how many people are infected versus how many tests were positive. You may have 1,000 positive tests, but if those 1,000 samples came from 500 people, it has only affected 500 people. So it depends on how the data are reported, and this is really becoming a big mess in the United States. And some public health departments are reporting RT-PCR data, and now some of them have started reporting serology results as well. That also leads to quite a bit of confusion as to what is the number of cases that we have to be we are contending with. Of course, WHO recommendation is quite clear. It just came out, I think, a day or two ago. They expect COVID-19 positives should remain less than 5% in a given population for a minimum of 14 days for all activities to resume, quote unquote, in a new normal way. So that is a very important guidance that the WHO has provided, because so many governments around the world are struggling as to when can we reopen the economy, when can we reopen the colleges, when we can get back to quote unquote our normal life. And the guidelines clearly says that the RT-PCR data should be less than 5% for minimum 14 days for economies and other things to be rebooted or restarted. Can I have the next slide, please? So as of today, FDA has identified there are over 64, and I can tell you all these numbers will be inaccurate because there are so many things are happening so quickly. So 64 of the tests that are available in the United States as of this afternoon are molecular diagnosis, which is primarily RT-PCR, and there is only one antigen detection test that has been approved. And 11 antibody detection tests are available. And out of that, 10 of them are rapid, as Dr. Ravi mentioned. Less than 50% of them, the sensitivity is less than 50%. It's almost worthless. And so far, only one ELISA test has been approved. That was developed by Mount Sinai, investigators whom I personally know. And on this slide, deliberately, I have listed a number of very interesting websites. One is WHO, the other one is CDC, and the third one is Indian Ministry of Health. That updated a lot of information as of today. And the other one is the Johns Hopkins COVID-19 Resource Center, really spectacular website which provides you literally real-time information. And then there is another incredibly interesting website developed by Cold Spring Harbor Laboratories, and that is called BioArchive. BioRxiv stands for BioArchive. Please look at that website at least once a day if you're really interested in what's going on. What they're doing is they have made it a repository of all papers that are submitted for publication but not yet peer reviewed and not yet accepted. But they want that information made available to those of us who are interested in in understanding what's going on a daily basis. So that's a spectacularly beautiful archival site which really lists and publishes papers that are not even accepted and that have been just submitted. So I think those websites I would very strongly recommend people to keep an eye out on. Can I have the next slide, please? So currently approved therapeutics, as all of you know, chloroquine and hydroxychloroquine, which is an anti-malaria drug and often sometimes used to treat lupus, that was approved by what is called emergency use authorization by FDA. It was pure politics. It was approved simply because Our president, believed in it, and he bullied the regulatory bodies to give it an authorization. Now studies have come out, particularly from Brazil. It's actually dangerous to take chloroquine or hydroxychloroquine because more people who are on that drug ended up, suffering severe consequences of COVID-19, and also there was enhanced mortality. So there is no indication whatsoever scientifically as of now that chloroquine or hydroxychloroquine has any protective value. Remdesivir that was developed by Gilead, many of you know, Gilead is the one company that produced hepatitis C therapeutic. It's a 2-week regimen that actually cures people of Hep C. And that's the same company that developed remdesivir. This is the only drug that has been approved as of today by FDA. And again, this is also by emergency use authorization. And this basically reduces the hospitalization from 14 days to 11 days. And remdesivir is effective only if it is given early on or when the symptoms are mild. When the symptoms become very severe and the patient is pretty much in crisis, it has little or no effect whatsoever on the outcome of the outcome in terms of patient outcome. And then the next two hot items that Dr. Ravi again kept mentioning, that neutralizing antibodies. Patients who recovered from COVID-19 or SARS coronavirus 2 infection, who have high levels of neutralizing antibodies. It cannot be rapid test antibodies. It cannot be ELISA antibodies. It has to be neutralizing antibodies because biologically those are the only relevant ones. And in America right now, they're looking at a titer of 1 in 320 or 1 in 640. That means your serum can be diluted 320-fold, and still there should be a substantial amount of neutralizing antibody. Only those donors, people who have such high levels of antibodies, neutralizing antibodies, only though their plasma are being collected and will be used for passive immunotherapy. And of course, instead of using a whole convalescent plasma, some of the companies are now purifying the immunoglobulin fraction from the plasma so that it becomes more like an intravenous IG that many people use for many autoimmune conditions. And so those are the two hottest commodities right now in the United States. In order for us to be able to identify potential donors, we have to have neutralization assay to make sure that they have protective antibodies that we can give it to other people who might need. And there is another drug that has been approved, Propavan. Basically, it is for sedation for patients who are miserable in the hospital, and it has nothing to do with treating COVID-19. And there is a whole bunch of drugs that are being tested for repurposing, drugs that have already been approved for as antiviral drugs against many different viral infections. And there is some people are hoping that some of those drugs might in fact be beneficial. So that all those drugs that are being tested clinically are listed in that particular website. And those who are interested in the drugs that are in the pipeline, please go to that website and you will get a lot of information. Can I have the next slide? So the repurposing is being pushed because if you recall, remdesivir was not developed against COVID-19. It was developed against Ebola, believe it or not. And so it had shown beautiful effects in the in vitro experiments or in the cell culture experiment. It shut down Ebola replication. So they took advantage of that, and they were hoping, because it's an RNA-dependent RNA polymerase, it's a nucleoside analog, and that mucks up the protein. And they hoped that it might work, and apparently it has worked. I can tell you this is not a drug that I will depend on too much, but it does help. Because if you reduce hospitalization from 14 days to 11 days, that is of some benefit. But it's not a cure-all. It has a lot of problems. It has a lot of liver toxicity. There is many, many issues associated with it. But right now, that's the only drug that has been approved. Can I have the next slide, please? Okay, so this is incredibly important. Dr. Ravi already showed the graph that I'm showing you on the right-hand side. So look at the days since exposure when the number of cases are reported. So upon exposure, these are all basically contact tracing. And you look at the number of cases that you will identify by RT-PCR is by about day 4 is the peak, and between 3 and 5 days you have most of the cases, between 2 and 5 days. This is between 2 and 5 days post-exposure to a known index case. The number of cases happen, and you can see then it drops off after 6 to 7 days. So there are two reasons, at least for this. One is the sensitivity of the assay that Dr. Ravi already mentioned. The other one is some of these individuals might have cleared the virus very rapidly. Okay, and now look at the bottom curve also shows how rapidly, probability of the fraction of individuals who are going to get disease very early on versus as the time passes. Why is this important? What I would like to impress upon you is that, as Dr. Ravi mentioned, in this particular disease, the IgM antibody responses appears maybe 7 to 8 days post-exposure. And the IgG antibodies don't even show up for 16 to 17 days. So if somebody is cleared of viral infection within 5 to 7 to 8 or even 9 days, you can see why, although they are recovered patient, their sera cannot be convalescent sera because they will not have neutralizing antibodies of the IgG isotype. This is something incredibly peculiar about this virus. So there are two messages an immunologist gets out of it. Number one, the virus infection itself is self-limiting. Number two, the innate immune system, like macrophages, monocytes, neutrophils, they themselves must be controlling the viral infection very early on in most of the patients because the virus does not persist long enough to provoke what we call an adaptive immune response. So therefore, the primary viral infection is not serving the same purpose as a vaccine would suffer. Vaccine would provide you the protection, correct? We are all vaccinated with rubella, mumps, and measles and all that. Why do we, when we get exposed to the virus, we don't develop the disease? We don't even know we got exposed because the immune system remembers the previous exposure and very rapidly mounts an immune response. That is called an anamnestic immune response, or a very rapid immune response within 24 to 48 hours. Your antibody levels go sky high. Whereas in COVID-19, look what is happening. The virus gets much cleared almost a week ahead of the appearance of the adaptive immune system. So although people might have effectively cured confirmed COVID-19 infection, they may not have protective immune response. That is really one of the most interesting properties of this virus infection. So I really want all of you to pay attention to the timeline on this particular slide. I'm going to leave all of these slides with Dr. Sagar and he can share it. Karuna Sagar, feel free to share it with everybody. Can I have the next slide, please? So what are the projected timeline for treatment and prevention? You can see there are 66 programs working on 3 different approaches. Believe me, that number of programs has shot up to 84 as of yesterday evening. So there are, I already told you there is a number of drugs that are being repurposed. And if you go to that website, you will get a list of those drugs. And there are 16 trials with various antibodies, convalescent sera, hyperimmune immunoglobulin, human monoclonal antibodies, or antibodies that worked against SARS coronavirus, the previous SARS coronavirus. They are testing to see whether there is cross-reactivity. And as of today, there are over 80 different vaccines that are being developed. This slide is already antiquated. So I want all of you to know there's a massive effort. And the most exciting data appeared about 3 days ago. This is a brand new way of vaccinating people. It is nothing but messenger RNA of the virus that is encapsulated in nanoparticles. And this is the vaccine that was used, and this is produced by Moderna. And Moderna has gotten into collaboration with major pharmaceutical companies to produce over 300 million doses, by early next year. And this vaccine, after 2 doses of vaccination, the 8 healthy volunteers were tested. All of them developed pretty high titers of antibodies against COVID-19. So America is betting right now that this vaccine might be the first vaccine that will move forward. However, an Indian pharmaceutical companies are pursuing 4 different companies in India are pursuing the vaccine that was developed by the Oxford University in England. The Oxford University has much more rigorous test than the Moderna vaccine, and they basically vaccinated non-human primates, and they intranasally challenged them with very high dose of COVID-19, and not a single monkey developed, disease, post-vaccination. So that is showing the most promising result. And a very similar vaccine is also being developed in Wuhan, and they have very similar results like the Oxford University has. And the Indian companies, I know for a fact, they have partnered with the Oxford University to see whether that vaccine can be brought to India as rapidly as it's humanly possible. There are a couple of companies in Hyderabad and Serum Institute in Pune. They are the ones that are in the forefront of developing vaccine in India. Can I have the next slide, please? I'll try to go through that. So, pathway to vaccine development, everybody says, why can't we have a vaccine early on? I can tell you, typically, well, we have passed the exploratory stage, and luckily we have already passed the preclinical stage for COVID-19 vaccine. So let's not waste time on that. So right now, the clinical development, phase one, many of the vaccines have already undergone phase one clinical trials, and I told you about the Moderna vaccine, which is an mRNA-based vaccine. And now they are going into phase two, rapid phase two, as well as phase three clinical trials. And that's why typically, these kinds of things take anywhere between 8 to 12 to 15 years. Because of the pandemic situation, many rules and regulations have been laxed. And therefore, the vaccine clinical trials are going in an incredibly rapid pace. And phase two, I think, will be completed in next 4 to 5 months. And phase three, probably will be early spring next year. And of course, once that is done, the regulatory review has to take place, and you have to manufacture billions of doses of this. And of course, quality control and all of those issues one has to worry about. Please appreciate there are several challenges to vaccine. Number one, we need to show that the vaccine is efficacious. Number two, when you give it to hundreds of millions of people, safety becomes, all of a sudden you will begin to see problem that you may not see with 100 people or 20 monkeys. And therefore, safety becomes a paramount importance. Number three, once you have an effective and safe vaccine, please think of this. Now they have to bottle them into 7 billion bottles if it is one dose. If it is 2 doses, you have to do 14 billion vaccine bottles. And you need 14 billion syringes. And look at the manufacturing and all the other issues, transportation, storage. So therefore, when people think of vaccine, it's not simply development of the drug that is important, but how do we deliver those drugs to the masses in the numbers that we need to deliver in a pandemic condition when the supply chains are drastically disrupted. So I want all of you to appreciate the challenges associated with the development of the vaccine, which is listed here. But post-development, there are plenty of challenges in terms of starting with manufacturing to packaging to shipping to and actually implementing the vaccination program. So I want to draw your attention to some of those challenges. Can I have the next slide, please? Yeah, and so there are many different methods of vaccine development. I don't want to bore you. That's very apparent here. I talked a lot about the mRNA vaccine. Many of the other vaccines are recombinant glycoprotein that's on the surface of the virus. And some people are preparing what are known as virus-like particles. They look like virus-like particles, but they're not viruses because they won't have the genome inside them. And I have given you examples of the approaches that have been successfully used against which vaccines in the past. And of course, cDNA vaccines not a single vaccine has been approved for human use, but they're routinely used for veterinary vaccination purpose. And viral vector-based vaccine, that includes, for example, rabies vaccine and distemper vaccine. They're all built on adeno-associated virus or adenovirus backbone. And of course, inactivated vaccine that we have been using forever, polio and of course, rabies. Of course, oral drops is it's live attenuated, whereas we also know the Salk vaccine was, inactivated polio. Attenuated virus vaccines, we all are very familiar, mumps, measles, rubella, and oral polio. And the chimeric vaccine, basically, they developed it for Zika virus, and it was on a dengue virus backbone, and they put Zika virus surface protein onto that backbone to grow up the virus very rapidly and to prepare a vaccine for Zika. And of course, nanoparticle vaccines, I already told you that are being used. Moderna uses mRNA in combination with nanoparticles. And one of the vaccines under clinical trial is for flu. So there are many different types of vaccine products that are under development. And we are fairly confident we will have an effective vaccine. I wouldn't say next 3 to 4 months, but I would realistically next 12 to 15 months. Can I have the next slide, please?
This is the last one.
Oh, that's it. So why don't I stop there? And I think I've highlighted some of the opportunities and challenges that we will have to see. I'm happy to take questions at the end of the presentations.
So thank you very much, Prabhakar, and we hope we will have some question and answer session at the end. Now, I would like to invite Dr. Gangadharan, who is the professor of mechanical engineering and head of the Center for System Design and president of the Institute Innovation Council at the National Institute of Technology, Karnataka, which is based in Surathkal. He did his PhD from IIT Madras, and then he has been in different institutions, and he is presently with the NITK, and he is working a lot on medical devices. So we are happy to have Dr. Gangadharan, who will tell us about the technological innovations in India. So number of engineers have come up with novel solutions. So he will give some examples, and he himself is involved in many of these. Dr. Gangadharan, please.
Yeah, very pleasant good morning to everyone, and maybe for Professor Prabhakar, good evening. On behalf of NITK Surathkal, warm welcome, and from Team Center for System Design and the team which works with the COVID warriors at my lab, we take this as an opportunity, and the great challenge always throws great opportunities here. We saw how another species called engineers can work with doctors, the medical doctors, and it was so traditional that medical doctors and engineers were poles apart. And we were trying to work together and see how engineers can help in a pandemic situation. The problem here was all of us were shut down or locked down, and all the resources were scattered. So during that period, one of the activity which came to the forefront was the requirement for ventilators. Even though now things have changed slowly, but we had two approaches. We had a national team, pan-India team working on that. Then we had two approaches. One was to see whether an existing ventilator can be used for multiple patients by just using a simple ventilator circuit split for 2 patients or 4 patients. And we were very clear how to make the devices for that. But later we found out we just making it alone is not sufficient. We need to convince or interact with ICU experts, anesthetists, and respiratory experts. In that connection, we had a lot of interaction with SHEMA. Actually, KSEGDE Hospital, Dr. Vikram Shetty coordinated the activity, and Dr. Sumalada, Sripad, and Harish was people who did the testing for this part of India. We coordinated the testing, and they did this split of ventilator on artificial lungs as well as high fidelity mannequins. This was one work which we did. This work should never be used. I mean, we rarely work on an item wherein we say that this work should not be used, because we were hoping and praying that the number of patients should not come to a level where existing or available ventilators are not enough. So that was one interaction we had with doctors that had thrown open quite big challenges. How to convince my medical fraternity on what I am doing, and how do I communicate or make him experience the concept on which I am working. And then we moved on to ventilator design. At least there are 110 groups working on ventilator designs, and many of them may not see the light as a ventilator, or they may even end up in an ambu-bag respirator kind of a thing. But what I am interested in that activity is, now there is a trend. People from medical fraternity, engineering, manufacturing, and logistics working together, both supply chain, etc., working together. If this trend continues, this itself will be a great potential for us to make sure that we are in the right direction. So we found this as an opportunity, and working together with doctors and engineers, taking technology solutions. We are hoping this trend will continue, and we will have more than 80% of devices required for medicine or medical or healthcare being designed, developed, manufactured in India and used in India. Maybe we'll be exporting it. That's a kind of things which we have been seeing. Now, in order to convince my medical fraternity, what we are doing in engineering and how it can be viewed, we started looking at experiential learning. So I'll just put one of my window to show that how it was done. We found it was pretty difficult to convince people on when you want to say, yes, this is how we are going to do. I hope you are able to see my screen. So we started looking at, I'll just quickly show you how did I try to convince my friends sitting at a medical college. Of course, they were not with us. They were sitting at another location. If an ambu bag has to be pressed and released, how do I get the volume of air, the tidal volume required, etc.? Or if I'm looking at an mechanism which gives me an air or oxygen at required quantity. So these kind of things which we made, it was used for engineering. Now we are using it for collaborative activity with medical fraternity, wherein we are able to demonstrate what happens when some parameters are changed. So from here, we just keep moving, and without taking the technical activity here, we kept on explaining how this term, engineering term, need to be understood as a medical person. So this is something which we found that the online interaction as well as online experiential learning methods, both in medicine as well as in engineering, could be the way in which we are going to look at the collaboration. Now it's going to be always, I'm coming back to, I hope my screen has stopped. Yeah. This way, we started working with doctors. And what this is going to give us is a way forward to have global collaboration and a local manufacturing. So we look forward to work with medical fraternity and find local solutions for global problems. And the problems are not going to be from one domain. It will be a tough task to convince each other what we need and how we need to work. So we found this as an opportunity with that minimum inputs, because we are really running out of time. We have already reached almost 10 o'clock. We would like to see more questions and answers, especially when both Professor Ravi and Dr. Ravi and Prabhakar talked. I felt a little more comfortable with COVID because the way in which they were looking at things were really good, and even a non-medical practitioner or person could understand it so well. So that clearly indicate the depth in which they know. Because if I know things well, I'll put it in a simpler terms. So thank you very much for that. And with this, I will conclude saying that the opportunity given by this COVID and lockdown is a global collaboration with multiple species, engineers, doctors, manufacturers, designers put together to find local problem solutions and making it in local. With that, I conclude. Thank you for this opportunity. Back to Colonel Sagar, sir.
Thank you very much, Dr. Gangadharan, for this very interesting presentation, and I hope there will be more collaborations coming up as a result of your introduction to this collaboration. Now I would suggest that we take up some questions, and I think Dr. Indrani had some questions that were for Dr. Ravi. Dr. Indrani?
Yeah.
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 There were some questions to Dr. Ravi. Yeah.
Oh, the questions have come. Of course, there are many questions, but I have, due to paucity of time, just a couple of them have been picked up for Dr. Ravi as well as for Dr. Prabhakar. Dr. Ravi, as regards the high-end tests that are being conducted, several people have been asking whether Atmanirbhar program of the government will also take care of the consumables that are being imported for the nucleic acid and other tests? And a second question from another participant is, have you looked at indigenous kits versus imported kits in their performance?
The answer to the first question is yes. The Make in India initiative has led to a huge drive among Indian companies to make things like RNA extraction kits. We keep getting requests for evaluating indigenous RNA extraction kits. Most of them are column-based, just like the Qiagen extraction kits. Whatever few we've evaluated have done fairly well. We have given them our feedback. The answer to the second question, have you evaluated Indian versus other kits? In the last 60 days that we have been doing testing, we have had experience with 10 different RT-PCR kits. In fact, currently as I'm speaking, we are doing an evaluation, comparative evaluation of these 10 kits. 3 of them have been manufactured in India. And they are from my lab. They are from Altona, which is a Gujarat company. And the third one is CRISPR-Cas9 one, which has been developed by IIT Delhi, which we have just started evaluating. So I will have the results probably in a week's time. But one thing I have learned personally is there is no gold standard in RT-PCR. So what we have done, we scratched our brains, and what we have done is we have sequenced full-length SARS-CoV-2 in close to 49 patient samples. So we are taking sequencing as the gold standard. We know that viral sequences are present in these samples, and it's full length, and it has all the genes. So that is by default what we are taking as gold standard to look at the comparative evaluation of these 10 kits. So hopefully by next week we will have these details.
Thank you. What is your opinion on cartridge-based equipments or test kits? Cartridge-based test kits.
Yeah, there are 3 actually in the market. One has still not been popularized in India, and that is the Abbott one. But India has experience with the CBNAT, which is the Cepheid-based TB machine for which SARS-CoV-2 kits has come. And we have, of course, our own indigenous TrueNAT, which is now become even a confirmatory test with a second cartridge. The limitation with cartridge-based tests is that the numbers you can do is very, very less. If you are talking of use in public health or on a large scale for clinical facilities, it's a physician's desktop test. Very convenient. If you have 1 or 2 patients, you have 10 patients, you can do the test. They are faster, and from biohazard point of view, they are much more safer than RT-PCR, because at least the TrueNAT has a lysis buffer into which directly the sample is collected. So there's no exposure of the laboratory personnel to infectious virus. So these are the pros and cons of the cartridge-based test.
There is one more question that has come, and that relates to the infectivity of people, the caretakers of patients in hospital. It is alarming. In spite of using PPE, they are getting infected. Does this reflect on the quality of the PPE or the sensitivity of the test?
Why people are getting infected in healthcare facilities? One reason already people are asking is it the quality of PPE? Honestly, it's a very difficult question to answer. I suspect it must be the quality of PPE in a huge outbreak when there's an acute shortage of PPE. So quality takes a big beating. That's number one. But I personally know there's another factor which people don't acknowledge, and that is breach in protocols while working in PPE. There are two reasons for it. People should not work more than 6, max 8 hours, but people work 12 hours. Dr. Prabhakar will tell you that in New York and in some of the hospitals in the US, people have worked 14 to 20 hours because there's an acute shortage of manpower. The longer you work, the tired you get, you make mistakes, and you breach protocols. Which is one of the major reasons that people get infected.
Thank you very much. Now we have a couple of questions for Dr. Prabhakar Bellu. I know we're running out of time, that's why we've just chosen very few of them. Dr. Prabhakar, everyone has been talking about the IgM and IgG antibodies, realizing that the mode of entry of this pathogen is via the nasal tract, aerosols or whatever. The secretory antibodies would play a possible role. And therefore, when you think of vaccine, and you have spoken about the nanoparticle-based vaccine, do you look at local application to evoke a response that can prevent the very attachment during entry?
Yeah, no, that's an outstanding question, actually. So people have been struggling with that, whether the vaccine should provoke an IgA antibody response or a mucosal immune response, and therefore what should be the route of immunization. So if you look at all the respiratory viruses against which we have very effective vaccines, rubella, mumps, measles, they're all transmitted, via, nasal route. And yet, we have incredibly effective vaccines against them. And so therefore, some people are arguing, I don't think we should spend a whole lot of time worrying about whether it's IgG or IgA, as long as the patient has high levels of neutralizing antibodies. Please understand, IgG can get into every part of your body fluid. Whether it's a nasal secretion, you will have IgG, you have sera, you will have IgG, you have body fluids, you will have IgG. So IgG can be, present, it's omnipresent in the body. Unlike IgA, which predominantly hangs around in the mucosal surface, IgG can hang around on the mucosal surface as well as everywhere else in the body. And therefore, the global bias right now is in terms of eliciting a very high-titered, rapidly rising IgG and neutralizing antibodies. Literally, that's what being done. And if I can take 30 seconds and go back to what Dr. Ravi said. Dr. Ravi, what we are doing right now to understand the natural history, believe it or not, we don't even understand the natural history of COVID-19. Because these random reports, everybody has uncontrolled studies and rapid publication, this and that and whatnot. What we have chosen in our medical school is to follow 60 COVID-19 infected patients. And we are going to collect 9 different time points, nasopharyngeal swab. And we are planning to do oropharynx as well as both, nares, the same swab. And so we are collecting all the samples to RT-PCR, of course, in the pathology lab, but as well as in our regular molecular biology lab. And we are doing qRT-PCR, followed by whole genome sequencing and virus isolation studies. Because we want to be able to correlate what are all these things measuring, exactly, and how do they relate to each other. That is one aspect on the virology side we are doing. And the reason we are sequencing the sequential isolates from the same patient is because we want to understand whether the virus evolves within a single patient, because we really don't have that. And also, does the virus evolve within a community? So those are the 2 other questions that we can answer with the sequence data to see whether there is nucleates arise as a consequence of ensuing immune response. The second part of the study is we are going to do ELISA, both for IgM and all subtypes of IgG, and we are going to do virus neutralization test because we have BSL-3 facility. Yeah. So I'm an immunologist. My bias is, can I develop a highly correlative ELISA data, that correlates, almost 100% of the time with the neutralization data. Is there an algorithm that I can develop so that not everybody has to do neutralization,
Exactly.
but they can use the algorithm to be able to, rapidly test. So it will serve multiple purpose. We will understand what the herd immunity is from a public health point of view. It will help us identify potential convalescent plasma donors. And number 3, it will also help us understand how protective it is and how long these antibodies last. So
Exactly.
I want you to know we are involved in this very massive program following 60 patients. Plus, we are looking at 40 analytes for cytokine, chemokine. Everybody talks about cytokine storm. We have no evidence for it other than people screaming. So we are looking at 40 different analytes, and we are looking at 27 markers on various monocyte, lymphocyte population to understand whether it's Th1, Th2, Th17, what is the role of innate immune system versus adaptive immune system. So we are looking at T-cell activation markers, T-cell proliferation markers. We are looking at T-cell exhaustion markers. I want you to know, hopefully, in next 6 to 8 months, we will have an unbelievable volume of data on 60 patients, and we are calling it natural history of COVID-19.
Great. That's really great to know. And
Yeah.
One last question.
studies of your
Yeah.
One last question, Dr. Belur. These respiratory pathogens are being addressed through injectable vaccines.
Yep.
There is a question which is asking whether intranasal route would be preferred to parenteral route for administration, because the receptor binding domains are located on the mucosal surface, of course, many areas in the body, but predominantly when you take oropharyngeal swab or you are taking the nasal swab, you are admitting that the viruses are expected to be probably located there. And therefore, is it not better to think of a vaccination by intranasal route to prevent the primary portal of entry?
Yeah. So, I mean, I think I partly answered that, but let me take it again. So there is the as of today, the most detailed description of 11 cases of COVID-19 was in Nature Medicine. That paper appeared about 2 weeks ago. They have done everything that one can do with a patient in terms of laboratory analysis. What they have found in running, although in the early stages everybody is thinking of it as because of the acute respiratory distress, everybody is thinking of COVID-19 as a respiratory disease. But now it is becoming loud and clear that it is actually a microvasculature and microclotting disease, and therefore it's buggering up literally all the capillary beds, and therefore you're beginning to now see, because the respiratory distress, what is happening is it is so acute, the patients go into crisis and some of them die and some of them recover. But the so-called recovered patients are really going through all kinds of cardiovascular sequelae is becoming a phenomenally important consideration in this disease. And there has been a beautiful study that just came out where they looked at 9 patients with influenza-induced, flu-induced ARDS versus COVID-19. And what is happening is that it's not the lung epithelial cells that the COVID-19 is destroying. It's actually vascular endothelial cells that it is attacking and destroying. And therefore, it is morphing gradually into a vascular disease rather than a respiratory disease. But the respiratory manifestations are becoming the most prominent ones because that's what is causing the acute distress. But if you look at it now, the renal problem is very rampant. The cardiovascular problem is now we can see in children, it is manifesting itself like a Kawasaki-like disease. There are over 115 cases in New York already. So therefore, that is one argument I'm making. Therefore, to think of this as only limited to respiratory tract is a fallacy, because you can readily isolate virus in the blood, you can readily isolate virus in the urine, you can readily isolate virus, believe it or not, even in the feces. So therefore, the virus has a predilection to go wherever the ACE2 is. Please, ACE2 is not limited to respiratory tract. ACE2 is an enzyme that is expressed in many different parts of the body, particularly the renal system. Please remember, this is a very important molecule in the renin-angiotensin, blood pressure control mechanism. And ACE2 acts, counteracts the ACE. the ACE2 action is to counteract the ACE action. So therefore, we are beginning to understand how this receptor function itself is being hijacked by the virus on one hand. And as a consequence, not only we are suffering the consequence of the viral infection, we are also beginning to suffer the consequence of loss of ACE2 function. So it's really, this is a rapidly evolving disease. So that is number 2. Number 3, I already told you 2 major non-human primate direct viral challenge experiments have been done, one in Wuhan and one in Oxford. And in both cases, 9 monkeys were vaccinated twice, and they were intranasally challenged with COVID-19 with pretty high doses. And if it was the respiratory route of vaccination was required, then these monkeys would not have survived, with no symptoms whatsoever. And therefore, although intranasal vaccination might turn out to be more effective, but there is no evidence that it is required at this point.
Yes. One last question from me, that is my own question. So now you have talked about the virus being shed through fecal samples, if kidney is affected, maybe also through urine. Having said that, transmission of the virus through polluted waters, therefore, becomes an issue in a country like India or in crowded cities where the treatment of the water may not be perfect enough. And therefore, taking on from where you said there could be other modes of entry of this virus.
No, I didn't say that. The virus is shed everywhere. The primary, there is no, actually this question has been actually addressed. There is no evidence for oral fecal oral route transmission as of now. But who knows? So when you have billions of people exposed, the virus may adapt itself. But right now, the primary mode of transmission of this virus is aerosol. It's as simple as that. Right now, there is no evidence that it can be transmitted any other way. Remember, original SARS coronavirus is a select agent, but COVID-19 is not a select agent. Most of the work other than work with the live virus is still allowed in BSL-2 conditions, including the preparation of samples for RT-PCR is done in BSL-2. And only if you are working with live virus, like virus neutralization or virus isolation, you need BSL-3 facility. That's all. So, and whereas the original SARS is listed as a select agent, and this one still is not listed as a select agent. So therefore, there is some peculiarity about the transmission of this, this virus. We will know. I think a lot more will be known in next 3 to 4 months. I can assure you that, because so many attacks are being conducted.
Thank you very much. I apologize all the people who have signed in and have had a lot of questions that we could not take up due to paucity of time. As of now, we've already overshot, but fortunately for us, both Professor Prabhakar Bellur as well as Professor Ravi were available, and therefore I want to thank you both for having at least answered a few of the questions that came in. Thank you very much. Over to Karuna Sagar.
Sagar, can I suggest something? Can you if there are a lot of questions, or maybe on your website you can create a Q&A, and if they pose the questions, as and when I have time, I will answer most of those questions. And Dr. Ravi, if you can take some time to do that, then we can leave this as a Q&A on your Nitte website. If you already have the questions. It's up to you.
Yeah, thank you very much. And I think they're a very good idea. So we can create a discussion forum in our website where people can put in questions, and we can pass on the questions to you for the answers. And of course, you will share the slides in case people want, and your email also.
One request to Prabhakar.
I look forward to meeting Ravi in Bangalore when I'm there next time. Ravi, I'll come to one in hands.
Please do. I also look forward.
Prabhakar, hello.
Hey, sir.
It was very interesting talk, and I request you offered to answer some of the questions which has been posted. If we can, your continuous 30 to 40 minutes, 45 minutes lecture can be beamed to our medical students, particularly medical students, postgraduates, undergraduate students, and even the staff. I think this will be very interesting and it will be very useful. I request India Karnasagar to organize one maybe continuous, it maybe can be interactive or it can be continuous lecture which can be beamed to our New Learn program.
I'll be happy to do that.
Thank you.
Sir, Gangadhar Nimmanost, very nice meeting you, Dr. Gangadhar. And I love to interact with you and I want to hear of your ideas.
Yeah, sure. We will maybe communicate through email because I knew the time is running out. Hence, I made sure that I did not speak much because whatever we are doing, because the time was very precious because I know other doctors may have to go for their work and the things of that kind. So I restricted. So we'll have offline interaction sometimes.
Yeah, thank you. I look forward to that.
So with that, I think we will try to conclude this. So I would like to thank all the resource persons for the excellent information that you provided and also the offer to continue the discussion through a discussion forum through the Nitte website. And of course, it's already late night in Chicago, but still Prabhakar stayed back to answer all the questions. And also I'm very sure Ravi has so many pressing engagements and he must be having several calls in between, but he stayed with us throughout to talk to us and answer the questions. So thanks to both of you. And also thanks to Dr. Gangadharan for being here and also offering the collaboration and to continue to work in this area. I would like to thank you also and thank to our Vice Chancellor and all the participants who have logged in, and we would like to continue the interaction with all of you. Thank you very much and have a nice day.
Yeah, thank you very much.
Good night for Dr.
Thank you very much.
Good night, everyone. Good morning, everyone.
Is the head of all this.